Welcome to the Cohen Veterans Bioscience webinar series. This is a monthly program sponsored by Cohen Veterans Bioscience for the brain research and systems biology communities. Cohen Veterans Bioscience is a nonprofit research alliance whose mission is to transform research in veterans' mental health through translational research. We're focused on understanding the molecular underpinnings of disease, such as PTSD and TBI, to develop better treatments and diagnostics. Before I introduce today's speaker, let me walk through some logistics. This program is being recorded and will be archived in the Cohen Veterans Bioscience YouTube channel and website. The presentation will be approximately 45 minutes in length to allow for a Q&A afterwards. Please use the Q&A feature on the website to post a question, and we will pose the question to the presenter. Today, we're delighted to introduce our speaker, Dr. Retsina Meyer, who is the Business Development Manager at Cohen Veterans Bioscience. As a translational neuroscientist, Dr. Retsina Meyer's work spans from human behavioral genetics and psychiatric illness, where the scientist investigated the role that dopamine genetics plays in clinical presentation of ADHD, to electrophysiology of learning and memory and cognitive function in awake-behaving animals, from mouse to rat to non-human primate, to the neuroendocrinology of the transition from acute to chronic stress. Dr. Meyer has worked in the field of translational neuroscience for over 15 years, and in that time has worked with numerous disease model systems, from epilepsy to aging and a variety of putative PTSD models. Retsina has leveraged methodologies both innovative and classic, established and completely novel, having developed new assays in the exploration of cognition and working memory. She has produced over 40 articles, abstracts, and invited lectures. Her academic discoveries gained international press, earned her a speaking engagement at the local TEDx conference, and served as the instigating discovery to found Resilience Therapeutics, a startup focused on novel treatments for PTSD and other brain diseases. As an entrepreneur, Retsina is an alumna of MassBio's Mass Connect program and Springboard's Life Sciences Accelerator, and is the 2014 champion of the International One Start competition awarded by GSK's SR1. She served as a science advocate and worked with the Science Policy Initiative, performing outreach, giving lectures, and meeting with members of Congress to appeal for science and scientific funding. She obtained a neuroscience PhD degree at MIT, where she earned 5 named fellowships, was a Presidential Fellow, Imperial College of London Global Fellow, and a Graduate Woman of Excellence. Prior to her work at MIT, Dr. Meyer was a Fulbright Scholar, serving as a representative of the U.S. to Norway, where she worked in the lab of 2014 Nobel Prize winners Dr. May-Britt Mosier and Edvard Mosier. She holds a bachelor degrees in applied mathematics, biochemistry, and molecular physics and biopsychology from the University of Arizona, where she was a Bayer Scholar, and worked in the laboratories of Dr. David Rowe, Dr. Carol Barnes, and Dr. Bruce McNaughton. Dr. Meyer has officially joined the CVB team and is excited about helping to realize the Cohen Veterans Bioscience vision and mission of accelerating diagnostics and therapeutic development for PTSD and TBI. Last year, Dr. Patricia Kabitzke introduced animal models in her webinar called Animal Modeling 101. What are the animal models and why are they used in research? In Animal Modeling 102, Dr. Meyer will build on the previous general overview of animal models with a focus on the frameworks with which translational model systems of post-traumatic stress disorder can be developed and assessed, leveraging the clinical literature. We will survey existing models that putatively model aspects of PTSD phenomenology and how Cohen Veterans Bioscience is working to advance our understanding of the clinical utility of these models and tests. It is my pleasure to introduce Dr. Meyer.
Thank you for that kind introduction, Michelle. As she said, I am part of the team driving the preclinical programs here at Cohen Veterans Biosciences. At CVB, our goals are to develop translational tools that will accelerate diagnostics and therapeutic development while also informing the underlying biology of PTSD, TBI, and other brain trauma disorders. Preclinical animal model systems are imperative in accomplishing these endeavors. In recent years, however, the field of translational neuroscience has become increasingly aware that successful findings observed in the preclinical realm have failed to translate to successes in the clinical setting. How might we bridge this gap in translation? Today, I'll review some of the frameworks with which to view the model development process, as well as some case studies. Over consortium meetings regarding how to mitigate the probability of failure of translation from preclinical to clinical discovery in psychiatric drug development, meeting members have lamented the failure to communicate across domains due to a lack of common language. That is why we have made this talk domain agnostic, taking a moment to introduce core concepts necessary to engage all stakeholders, as well as the concepts and questions that shape the thoughts and frameworks with which we can view animal model systems. At the end of this talk, all attendees should have a broad understanding of these major frameworks and the assays through the lens of clinical literature. I will begin with reviewing the main takeaways from Dr. Kabitzke's seminar, which you can find on our YouTube channel. It is an excellent introduction, and I highly recommend it for all audience members. I will then briefly overview the drug discovery and development process, and note where animal models may play a critical role. Then I will look into how clinicians and researchers approach PTSD, so that we can build on the survey of existing animal models that putatively model these aspects of PTSD phenomenology. And finally, I will close with how Cohen Veterans Bioscience is working to advance our understanding of clinical utility of these models and tests. In Animal Models 101, Patricia posed the question, what are animal models? An animal model is a living organism in which normative biology or behavior can be studied, or in which a spontaneous or induced pathological process can be investigated, and in which the phenomenon in one or more respects resembles the same phenomenon in humans or other species of animals. Why might we use animal models in research? Due to our common ascent through evolution, animals and humans and other have a preserved homology. That homology is represented anatomically, genetically, in developmental and metabolic processes. How are animal models generated? Animal models are generated in a number of ways: pharmacological interventions, electrical and lesion studies, genetic or transgenic animals, as well as neurodevelopmental manipulation. Animals have been useful and are used throughout science. In discovery, we've used them to explore disease mechanisms, confirm hypotheses, explore phenotypes and fundamental pathologies, and understand disease pathogenesis. In drug development, we use animals to investigate safety and efficacy, target validation, pharmacokinetics, and pharmacodynamics. The graph on the right shows the use of animal models in behavioral research from the 1960s to today. And you can see that this is a strong effort. Now I'll overview the drug discovery and development process, observing the failures in the pipeline and how animal models may play a role in mitigating these failures. Pre-discovery is where a potential therapeutic target can be identified. This can come anywhere, from a government-sponsored human genetic study to an academic animal program that identifies a potential factor that plays a role in a disease or set of diseases. That pre-discovery data is then elevated for research and target identification. In this phase, scientists search for chemical and biological substances that target these biological markers and are likely to have drug-like effects. Scientists use a variety of techniques to identify and isolate individual targets and learn more about their function. On the chemistry side, they search for compounds that will engage the target in a number of ways in order to find the ones that might be helpful in the treatment of a disease state or used as a tool to understand more about how that target works. Next, target prioritization and validation seeks the targets that are most likely to be useful in the development of a treatment for a disease. This is done by observing how the target engages and changes the biological substrate. Compounds are tested to ensure target engagement and the desired change. We advance to lead identification, where a lead compound or substance is one believed to have the potential to actually treat the disease. Leads are sometimes developed as collections or libraries of individual molecules that possess properties needed in a new drug. Testing is then done on each of these molecules to confirm the desired effect on the drug target. During lead optimization, individual compounds in these libraries are compared to each other, looking for the safest and potentially most effective medicine. This will include in vitro work as well as in vivo work to observe how the compounds are metabolized and the effect they have on the body of research animals. During preclinical development, an investigational drug must be tested extensively in the laboratory to ensure that it will be safe to administer to humans. Testing at this stage can take from 1 to 5 years and must provide information about the pharmaceutical composition of the drug, its safety, and how the drug will be formulated and manufactured. This is in preparation for the administration to the first-in-human subject. Pharmacological testing determines the effects of the candidate drug on the body, while toxicology studies are conducted to identify potential risks to humans. In the U.S., results of all the testing must be provided to the FDA, the Food and Drug Administration, in order to obtain permission to begin the clinical testing in humans. Out of every 5,000 to 15,000 new compounds identified during the discovery process, only about 5 are considered safe for advancing to human testing in human volunteers. At this point, the development company will file an investigational new drug form with the FDA to get approval to start a clinical trial. They will also seek IRB, Institutional or Independent Review Board, approval for that study protocol. Clinical testing is usually described as consisting of phase clinical studies, phase 1, 2, and 3. In each successive phase, increasing numbers of patients are tested. Phase 1 studies are first in human. They are designed to verify safety and tolerability of candidate drugs in humans and typically take 6 to 9 months. They will involve a small number of subjects, usually from 20 to 100 healthy volunteers, who take the investigational drug for only a short period of time. Testing will include observation and careful documentation of how the drug acts on the body, how it is absorbed, distributed, metabolized, and excreted. In phase 2, if safe, the compound will advance. These are designed to begin to determine the effectiveness and further study the safety of the candidate drug in humans. These generally take 6 months to 3 years, and testing will involve several hundred patient volunteers suffering from the condition that the investigational drug is designed to treat. Dosing will also be determined at this time. If successful in phase 2, with an excellent safety profile, good tolerability, and evidence of efficacy, the candidate compound will advance to phase 3. These studies provide expanded testing of effectiveness and safety of the drug, take 1 to 4 years, and involve several hundred to thousands of volunteer patients. The new drug application is the application to the FDA to market the new drug. It traditionally occurs after a phase 3 trial. It requires safety and efficacy from each stage of development, and obtaining approval may take 6 months to 2 years. After the drug enters the market, it may be involved in post-approval studies, such as phase 4. This may be useful when looking at new age groups or patient types. It may also compare the long-term effectiveness or cost of the drug to other marketed drugs available to treat the same condition. Some studies focus on previously unknown side effects or related risk factors. As with all stages of drug development testing, the purpose is to ensure safety and effectiveness of the drug. After 3 to 6 years of further clinical testing in patients, only one compound, on average, is ultimately approved as a marketed drug for treatment. So you can see that there is a lot of attrition in the drug development pipeline. Here's a brief summary of the probability to transition from one phase to the other. At the top is the probability of success. On the bottom, you see the probability of failure. In red, you can see where we have large percentages of failures. And that's the jump from preclinical to phase 1 to phase 2. So the jump from animal to human and from safety to efficacy. So let's dig in and see why some of these might fail. In 2014, AstraZeneca analyzed why their programs did not transition at these 3 major inflection points. The preclinical to phase 1 failures are primarily due to safety issues discovered when compounds advanced from in vitro development to in vivo animal models. Few decisions to not continue program are based on efficacy at this time. The next phase, even with a large attrition due to safety in the animals, a large number of phase 1 to phase 2 failures are also due to safety. PK, PD, pharmacokinetics and pharmacodynamic profiles begin to play a role, and those that look at in the studies that look at efficacy endpoints, which is not required at this point, you begin to see red flags. The phase 2A to 2B failures are primarily due to efficacy. So in the safety failures, if we're looking at the CNS drug development, we see that the failures often happen at the clinical stage. These speak to both compound and target safety issues. So on the right, you can see that not all of the drugs are having safety issues due to what they're targeting, but due to some other effect. So whether it is engaging in another target, or whether it's metabolized into something that is intolerable to the human. Due to these trends, the FDA is introducing a new type of trial, a phase 0 clinical trial, as part of its new critical path initiative. This is an effort to modernize and speed up our clinical trials process. In the phase 0 trial, the goal is to establish whether a compound will work like you expect it and want it to in humans. The theory is that such a trial would also quickly weed out an ineffective drug. So a subtherapeutic dose would be given to show whether the drug's pharmacokinetics and dynamics warrant continued exploration. The researcher would be able to tell that if the drug enters the bloodstream as it should, and whether or not it is interacting with enzymes as anticipated. This process will not demonstrate a therapeutic effect, nor would it replace the dose escalation, safety, and tolerance studies required in phase 1, but it's a potential way to mitigate some of these safety failures. Several programs ended due to efficacy were found to fall into 4 major categories. 40% were ended due to lack of data demonstrating a clear linkage of the target to the disease, or a lack of access to well-validated animal models of disease. Programs in this category may be considered to have a poor understanding of the role of that drug target in the underlying disease pathology. It can be thought of as a good failure in that there's little doubt that you've actually tested the hypothesis you meant to test. You just happen to be proven wrong. 29% of programs that ended due to efficacy were due to properties about the compound. These compound properties may have limited the dose, or it may not have penetrated the relevant target tissue. This also reflects concerns associated with the compound which limit human dosing. For many of the projects in neuroscience, there's no way to determine target engagement or pharmacodynamic activity. A portion of efficacy failures are due to trouble identifying the optimal patient population based on scientific understanding of disease. This suggests a failure to rigorously pressure test the biological hypothesis of the project. Finally, some projects had a lack of sufficiently robust data in the previous level to have advanced to this level. These failures would be considered outside the realm of science, more likely due to corporate governance and departmental incentives. For the failures related to science, animal models may help. They allow us to further investigate the target, potentially identifying informative translational biomarkers. They would permit us to strengthen the link between the target and the disease. They would allow us to assess tissue engagement, predicting PK/PD profiles in a clinical setting. Clearly, animals will help with certain safety profiles, allowing us to find safety margins in dosing, identify secondary risks, look at genotoxicity, and observe drug-drug interactions in commonly taken medication. They also allow us to investigate target liability. Animal models could potentially help in patient stratification as well, allowing us to find biological indicators identifying who would most benefit from the potential therapeutic. So we covered why animal models are used in general and how they can help mitigate drug development pipeline failures if used strategically. Starting with the clinical assessment of PTSD and moving swiftly to biology, we will examine how bidirectional translational research of post-traumatic stress disorder can develop and assess animal constructs by leveraging clinical literature. PTSD is a trauma-related disorder that can develop after a traumatic event. 70% of people will experience a traumatic event in their lifetime, and of those, 20% will go on to develop PTSD. PTSD symptoms include intense fear, avoidance reaction, alterations in mood and cognition, and hypervigilance or arousal. The Diagnostic and Statistical Manual of Mental Disorders, DSM, is the template on which diagnostic instruments are developed. There was a major shift recently from the DSM-4 to DSM version 5. There are now over 600,000 ways to have PTSD. However, the modifications were also clinically significant on another axis. Only 55% of those diagnosed with PTSD with the DSM-4 meet criteria with DSM-5. I'll let that sink in. Clearly, diagnostic criteria, which are meant to index disorders, will not capture the full array of symptoms seen in patients, and they're not informed by the biology. Additionally, with the fluidity of clinical assessments, discovery cannot rely on this framework for understanding the biological underpinnings of PTSD. So what might they use? Like I said, over 600,000 ways to have a PTSD diagnosis. Maybe what we're seeing as PTSD is not a single disorder, but a spectrum of disorders, like something you would see in autism, with similar but not identical biological subtypes. If we embrace this complexity, we can examine groups of patients beyond symptom level and get to the underlying biology. The NIMH's Research Domain Criteria Initiative, known as RDoC, concept was to rethink research of the psychopathology and do just that. Symptoms serve as the starting point. The framework would focus on biological systems. Genomic variants, certain brain circuit alterations, and even subsyndromic symptoms can cut across several pathologies. So then, rather than maintain these disease silos, the goal is to develop objective measures of psychopathology to create data-driven biological clustering of patient populations. At CVB, we're leveraging a systems approach to PTSD pathophysiology. We can make faster progress in understanding brain function and dysfunction across diseases by using this approach to redefine disease by mechanism across the human life cycle. The systems approach to understanding pathophysiology utilizes an interdisciplinary array of measures to develop hypothetical models for interpreting complex data sets. From these, researchers can investigate potential connectivity among assorted biological and clinical readouts, thereby developing robust hypotheses. This guides the transition from the descriptive methodologies, like the DSM, to a mechanistic approach of discovery, revealing principles of cells, cellular networks, organs, and emergent behavior, and their interactions across several spatial and temporal scales. By moving to the biological domain and examining how trauma and stress may affect many organ systems beyond CNS, central nervous system, and peripheral nervous system, going towards cardiovascular, neuroendocrine, and immune systems, we can better translate PTSD sequelae forward to the clinic and backward towards animal models. So currently, there's this mismatch between the endpoints used in those preclinical studies, behaviors, hormones, the endpoints used in basic human research, like psychophysics, cognitive assays, and those used in clinical trials, the DSM-derived self- or clinician-administered report. The RDoC and systems biology approaches aim to reconcile these three areas of R&D. We can further accelerate that progress initiated by utilizing these biological approaches by working specifically with the constructs that cross the translational divide. We're not currently able to ask an animal about feelings such as shame or guilt, but other behaviors, circuitry, and hormones are all measurable in both humans and animals. The top 20 translational constructs we found in the clinical literature. So these were the top 20 biologically or translationally relevant sequelae that were found in PTSD patients. So the major domains include behavior, cardiovascular sequelae, peripheral or circulating biomarkers, and of course the nervous system, both central and peripheral. Some of the constructs we identified as foundational, some phenomenological, but what we did see a lot of was relationships among these constructs. So they weren't quite necessarily independent. We found that those relationships spanned the domains, behavior, cardiovascular, and other hormones and things, and were tightly networked within those domains. We found that sleep appeared to be connected to just about everything. So let's take a look at the animal models that computatively model some of these aspects of PTSD, and then how CVB is working to advance our understanding of the clinical utility of those models and tests. I really enjoyed this quote from Stephen Zorn, CEO of Mind Immune, and it comes from the Institute of Medicine's book, Improving the Utility and Translation of Animal Models in Nervous System Disorders. We can learn much from animal model systems, but what we learn depends heavily on what questions are asked and how those questions are asked and how the results are interpreted. So first I wanted to get into assay model. So an assay is a means of quantifying a dependent variable. You can think of it as a test. It will be used to measure or assess the effects of various factors and experimental manipulations. A test should not produce a disorder. It would detect a symptom or a construct, but it wouldn't necessarily produce alterations in that construct. A model is a theoretical description of the way a system, process, or disease may work. It is not meant to be a complete recapitulation of a disorder. It's a simple representation of a complex system, reproducing or modeling only portions of the disease symptoms or constructs. So you can think an animal model induces a state change from healthy via over or underexpression of some biological variable, and the assay quantifies that disruption. So how do we find out if we are testing what we want to test? How do we ask these questions of our animal assays and our animal models? There's a number of questions we can ask to determine the validity of our animal work. So a question we may ask is, does the model mimic the molecular or anatomical basis of the disease? This would be construct validity. And if you have high construct validity, you would have very similar behavioral, neurobiological, and physiological responses as those observed in a high proportion of patients diagnosed with your disease that you're trying to model, in this case PTSD. In convergent validity, you might ask, does the model exhibit high correlation among performance patterns across the tasks or assays designed to measure the same neurocognitive process? So if you think you're testing working memory, you may want to see convergent results on water maze, radial arm maze, T maze, all these assays that are saying they're measuring the same thing. If you find differential effect in, let's say, the water maze and the radial arm maze, maybe it's due to a component of the test. So maybe it has to do with the coordination of swimming if you've induced a locomotor issue. You may be able to mitigate that by modifying the test so that they're engaging similar systems as well. So, for example, the radial arm maze has been used as a water version so that you have similar demands on the physical components of the task. Base validity. This is to what degree the similarity of the endpoints have to the disease-specific symptoms in human patients. So what degree of similarity does the readout from your assay or the modification your model makes on the readout of that assay have to this disease symptom in a human patient? So if you were providing a cognitive enhancer to a human, they may perform better on a working memory task. If you have face validity in your model, testing working memory in animals, you would expect a cognitive enhancer to increase their performance on the task. Predictive validity. Based on the currently available therapy, do the pharmaceutical treatments produce similar effects in both animals and humans? And so that last example would have predictive validity if both animals and humans perform better when given the same drug. Finally, and this is very important to us here at CVB when studying PTSD, etiological validity. Is the effect pathological and not merely adaptive? So would you identify a behavior as something out of the ordinary or abnormal and not a traditional way that an animal behaves? So one of the, to kind of go through these validity questions, I pose this question. Is floating during the forced swim test a valid endpoint? Meaning, is it going to help with the translational science discovery for your clinical trials? In the forced swim, the animals are placed in a cylinder and that they cannot escape, and they will swim, struggle, or float. So if animal A is floating and animal B is struggling, an interpretation, the major interpretation would be that the animal who's decided to float is giving up. He doesn't want to try to escape, and so he's helpless. whereas the animal who continues to struggle is hopeful. So he maintains this effort to try to escape even in an inescapable situation. Well, there are alternative interpretations of this animal behavior. For example, perhaps animal A has some reason to be energy efficient and feels the need to float because they're conserving calories. Alternatively, animal A could have some other physical characteristic which makes it easier to float than animal B. So you can imagine that a less dense mouse would be able to float easily, while a very dense mouse would sink to the bottom and therefore is forced to swim to live. Alternative interpretations of the struggling or swimming mouse is that it doesn't learn, or something that I've had happen in my own labs, that it likes to swim and it chooses to maintain swimming behavior. So how might we clarify what this behavior is getting at? What this animal is expressing by swimming or floating? Well, you could remove your compounds. So do you get the same results if you change the water temperature, alter the lighting? Again, are the animals similar in age, weight, sex, gender? There could be a background reason that some animals are more motivated to move and some are less motivated. But one thing that would be really desirable is if you're trying to use the forced swim as a test of depression or helplessness versus hopefulness, you might leverage other tests with an endpoint, such as that. So sucrose consumption or social interaction tests have also been used to study depressive phenotypes. Caveat though, we wouldn't necessarily consider this the most translational endpoint because you cannot study it in humans. The IRB has not approved. A successful bidirectional translational endpoint is pre-pulse inhibition. Briefly, pre-pulse inhibition is the automatic inhibition of the startle reflex, the response to an intense or abrupt stimulus that causes you to jump. It also causes animals to jump. When it's preceded by a weak lead stimulus. This is the pre-pulse. So PPI is an example of the ability to study the same endpoint in both animals and humans. PPI is diminished in a number of different neuropsychiatric disorders, including schizophrenia. It is not a complete recapitulation of schizophrenia. It's just a subtype of the symptoms that you can find. It has good translational validity because there is construct validity, meaning that the same synaptic circuit, that corticostriato-palidopontine circuit, is present in both the humans and the animals. And the disorders that demonstrate PPI alterations have later been found to have abnormalities within these circuits. It's also been shown that with a transgenic-derived model that has the Huntington's protein problem, has a similar behavioral readout as the Huntington's patients in PPI. PPI has face validity. The startle response is expressed and measured with almost the same equipment and software in both humans and animals. It also has predictive validity. Several pharmacological manipulations have been examined for their effects on PPI in healthy human subjects. These alterations in PPI produced by the drugs were similar in both subjects. In fact, in a pharmacologically induced schizophrenia model, PPI was altered with the administration of the antipsychotics, which normalized it. So at CBB, we were surveying the field and looking at what were some of the putative PTSD modeling systems. We found there's a number of PTSD models used in the field, and they span from processive stressors to developmental, environmental. We see comorbidity explorations in animal work, life-threatening sorts of models. And we have a lot of resources at our fingertips and tools to use. But how can we choose which tools to use? Well, firstly, we encourage the use of multiple models that employ different approaches so that you can combine that converging evidence along a measure or construct or assay. So again, to this slide, well, with the clinical constructs, we started with these 20 anchor constructs in the clinical literature, and we were aiming to see how those PTSD models may alter these constructs. We have multiple assays to explore each construct that in the animal work. And so again, we can look at, if we're looking at hyperarousal, we can look at their baseline startle, a fear potentiated startle, the PPI that we just discussed, threshold to panic attack induced by different compounds, which has been directly studied in humans as well. So we have many different assays that we can use to look at that same construct. In the clinical literature, we found that the three largest or most pervasive constructs with the most translational potential were startle, fear, and sleep and EEG. Again, we have multiple assays with which we can look at each of these constructs. It gives us many tools to use, and especially when we're looking at animal behaviors or animal readouts, gives us different ways to ask questions in case the model does not permit us to investigate a construct in a particular way. And what we're doing is similar to the approach of clustering patients by their biological signals in the RDOT. We're assessing these PTSD model systems across the constructs to start to identify clusters. We're utilizing these constructs and their representative assays and finding that models are having different clusters, even in the published literature. Some models will have multiple constructs. Others have only been explored in a few constructs. We're finding some gaps, and the information doesn't exist in the literature, so we've started to perform de novo research. We've taken the PTSD models, and we're putting them through the same tests, the same assays, so we can explore these constructs. This is the sleep, the biomarkers, the psychophysiological reactivity, cognition, and what we're looking for is how do these models cluster in those readouts and constructs. So if those constructs then also map to a particular patient cluster, what you might have identified is an animal model that you could use to screen your drugs, stratify your patients, and de-risk your program for a certain type of patient. So for example, if immobilization stress was giving you readouts in hyperarousal and fear constructs, while chronic social defeat was giving you strong readouts in depression and anxiety constructs, you would be able to see how that might map to patients who have those subtypes of PTSD, and then compounds that were appropriate for ameliorating the effects of immobilization stress would be appropriate for the corresponding patient cluster. So CVB is doing much more around animal models, and that is under our AMP It Up initiative. So AMP It Up stands for the Alliance for Modeling PTSD, Innovative Technologies, and Uniform Practices. AMP It Up is a collaborative working group launched by Cohen Veterans Bioscience to jumpstart the field of PTSD animal modeling, to harmonize constructs in human and preclinical animal work, and computational models to facilitate the development of translatable preclinical model systems, to develop best practices, standards for reproducibility and robustness of PTSD model development and study conduct, To explore innovative nano and imaging technologies, to expand the armamentarium for preclinical research, explore the use of computational modeling approaches to provide a complementary modeling approach to the use of animal models, to centrally collect, synthesize all available information about models, identify gaps requiring additional study, and support the study programs to address those gaps. So if you'd like to, I invite you to get involved today. Thank you so much for your time. I appreciate you spending your Wednesday lunch break with me. And Michelle, maybe we can open it up to questions.
Yes, thank you, Dr. Meyer. That was a very informative and engaging webinar on animal modeling. So yes, please feel free to submit your questions to Dr. Meyer. So there is one question. How will the RDoc and systems biology approaches improve upon current approaches?
So currently, like we were talking about earlier in the presentation, the current approach is using the Diagnostic Statistics Manual for Mental Disorders in order to diagnose patients. And so that's the clinical readouts that you're getting. And it's more of a descriptive way to look at PTSD. As we transition to RDoc and systems biology, we're really trying to engage a more biological understanding or mechanistic understanding. And when you're using therapeutics to treat a disorder, while the readouts might be those descriptive symptomatologies, what you're targeting with your compounds are actually those underlying biological mechanisms.
Okay, any other questions? Here's a question from some participant. When you spoke to the use of systems biology to redefine disease or disease symptoms by mechanism rather than qualitative descriptions, it brought to mind the concept of endophenotypes. How is the concept of constructs you described different from endophenotypes?
That's a great question, and actually something I had meant to touch on, so I think I might have a slide for you. Perfect. There we are. So constructs are very similar to endophenotypes. The concept is similar in that endophenotypes are not a recapitulation of a syndrome, but they're a piece of the syndrome. So their endophenotype is also known as an intermediate phenotype, some people might call it. Our constructs, likewise, are not recapitulating the entire disease, but only a piece of the disease that we're trying to study in this very biological way. However, different from the endophenotype, the idea of construct is agnostic to the relevance of genotype. So endophenotype, by definition, is trying to tie it back to a genetic component, whereas we take a genetic agnostic point of view when thinking about the construct, although genetics could be a very pertinent component of constructs as well.
All right, another question. You had mentioned PPI is a good bidirectional translational endpoint that can be measured both in animals and humans. Are there other endpoints?
Yeah, so any endpoint that you can measure without a grand leap of interpretation would be significant for bidirectional endpoints and something that you would maybe want to pursue and prioritize over other things. I think what comes to mind as one of the most compelling bidirectional translational endpoints outside of behavior is actually imaging the neurocircuitry. So in different PTSD patient populations, you'll have circuit dysfunction of arousal or emotional regulation. And we can now actually image animals in a similar way to the way we image humans and identify dysregulation in those circuits as well. So neural circuitry imaging, those sorts of endpoints I think would be really valuable.
Very interesting. Thank you. Any other questions from participants? Okay, well, I'd like to once again thank everyone for participating in the webinar, and just keep an eye out that this webinar will be posted on the Cohen Veterans Bioscience YouTube channel and website. And again, we hope to see you at the next webinar. So thanks so much for attending.
Thank you.